Well, hello, welcome to today's talk, which is part of TechFest. I'm really excited to be here to talk to you all about X-rays, what they are and how we use them to see through fossils and study them. So let's get started. First thing I'd like to do is introduce myself. This is me, I'm the one on the right. I'm a paleontologist. So a paleontologist is just a scientist that studies extinct life, and most often we use fossils to do that. And that includes the fossils of animals like my friend here, Frida. Now, I work at the Museum of Natural History in Oxford, pictured here. So this museum is part of the University of Oxford, and it was established in 1860 and includes much older collections as well as modern specimens. Now it's relatively small, but it has a really rich history and some incredibly important objects. So we have over 7 million objects in our collections, and these include rocks and minerals, insects and other arthropods, including some living ones, taxidermy animals, art, books, wasps' nests, skeletons, glass models, jewelry, letters. We have so many different items. And many of them were collected by quite famous scientists in the past. Now, each year, about 750,000 people come to Oxford to visit us, as well as hundreds of scientists and other academics who use our collections for research. Obviously, things have been a bit different this year. Now, one of our most famous objects is our dodo. So there's a reconstruction of the dodo in the top right of this slide. So the dodo is so special that it features in our logo. The reason it's special is because we have the only surviving soft tissue from a dodo, and that's the mummified skin and the head, which you can see here along with the foot and other bits and pieces. Now this specimen's over 370 years old, and the dodo, of course, as I'm sure you all know, is famously extinct. In other words, it no longer exists, so only its bones and this bit of soft tissue survive. Now the last dodo was seen alive in 1662, and it was definitely completely extinct by the 1700s. It's one of many animals that have become extinct over the last few hundred years due to human activity. So a lot of our visitors come to the museum to see the dodo, but they also come to see animals that went extinct or became extinct much longer time ago, way before humans existed. I am, of course, talking about dinosaurs. So when we think of fossils, most of us will think of dinosaurs like this cast of Tyrannosaurus rex, which is in the main hall at the museum. Now, T-Rex was, of course, a very distant cousin of the dodo and of all birds, because birds are the living descendants of dinosaurs. So as well as the fossils of big vertebrate animals like dinosaurs, we also have in our collections other types of animals, including invertebrate fossil animals like trilobites and ammonites, which I'm sure a lot of you have seen and possibly even collected for yourself. So these fossils in our collections are really important historically as well as scientifically. Now, one of the best examples of this is this fossil. Our museum has the first dinosaur fossil to be scientifically described, and this is it. It's a jaw from a theropod dinosaur called Megalosaurus bucklandii. Now, as you can see from the pointed teeth, this was a meat eater, and it grew to about 8 meters long from nose to tail tip. So this fossil came from a quarry near Oxford, dating from the middle of the Jurassic, and it was described in 1818 by a man named William Buckland. So Buckland is pictured here, and he is teaching a bunch of people about fossils, in this case, an ammonite. So Buckland was the first person to describe a dinosaur scientifically, and by describing it, I mean he wrote a scientific paper about what it looked like and where it came from, and with ideas about what this animal was. Now, in other words, he made an official record of it for other scientists to read and learn from. And this is how science still works today. We write scientific papers about what we've discovered for other scientists to read and learn from, or to argue with, because we don't obviously always agree with one another. But what a lot of people don't know about this first paper that Buckland wrote is that at the same time as describing Megalosaurus, Buckland also announced another new fossil, and it was something just as amazing. So that second fossil was this. It's another jaw, But whereas the Megalosaurus jaw was huge, this one is tiny. You can see the scale bar at the bottom, that's centimeters. So this thing is only 2 or 3 centimeters long. It sits easily in the palm of your hand. Now, this is the fossil jaw of an animal like this. It's a very small mammal. It's about the size of a rat. And it might not look like very much, and it might not seem very impressive compared to a dinosaur jaw, but for scientists, it was just as important, and in some ways more surprising. And the reason it was so surprising is that the rocks it came from dated to the middle of the Jurassic, and that's the time of other dinosaurs and giant reptiles. Now, 200 years ago, when they found this, scientists thought that the only giant reptiles lived in the middle Jurassic. So they didn't think that there were any mammals. So this fossil came as a real shock. It was the first evidence that mammals, which is the group that we belong to, that feed their young on milk and have fur and warm blood, they actually lived at the same time as the dinosaurs. And it totally changed our understanding of the evolution of mammals. And it's ancient mammal fossils that I study. So I'm an early career Leverhulme Research Fellow at the museum, and I'm interested in understanding the origin and evolution of mammals. So basically, where we all came from. Now, as a paleontologist, I'm interested in studying their fossils. And a lot of people think that mammals only appeared and evolved after the dinosaurs went extinct. But their family, our family, is actually much, much bigger, and it stretches back much further than this. So the ancestors of today's mammals lived alongside the dinosaurs about 150 million years ago in the Jurassic. Now, before that, in the Triassic, there was also the close relatives of mammals called cynodonts, when we evolved from among them. Before them, even further back in time, over 252 million years ago, there were animals called therapsids and synapsids. Now, these are our distant cousins, and all of these animals belong to our family tree. So our evolution has actually been taking place over the last 350 million years, but we don't really look very much like our ancestors. So do you remember Frida, who we met at the start of this talk? Well, she's actually one of our ancient cousins who lived 265 million years ago. Now, how is it that we could be related to animals like Frida when we look so incredibly different? Well, that is an important question. And to understand how the bodies of animals have changed over millions of years, how they've evolved, we have to study their skeletons, which are preserved as fossils. Now, one of the places that I search for fossils is on the Isle of Skye. That's on an island on the west coast of Scotland. Now, as a paleontologist, I'm looking for the fossils of mammals and other extinct life in order to understand more about how they looked, how they moved, how they were related to other animals, and then, of course, this bigger picture about how evolution works across millions of years. There are many different ways to be a paleontologist. But for many of us, our job involves traveling to a fossil locality, like I do, to discover bones. And then we collect those bones and we bring them back to the laboratory. Now, sometimes paleontologists study fossils that were collected in the past by other people, and they're already held in museum collections, such as the museum I work in. So once we have our fossils, we examine them. We perhaps use microscopes to look at wear patterns on their teeth, or we analyze them chemically for isotopic signatures. We measure them, we compare them to living animals to understand more about them, and we collect other data. Now, people imagine that paleontologists probably, spend most of their time out in the field collecting fossils, but actually we spend most of our time on computers. We're gathering data, we're coding, and we're writing. And we also spend a lot of time working with friends and colleagues to understand what it is that we're seeing. And then finally, we write about it. So we publish papers telling everyone what we've discovered, just like Buckland did 200 years ago. Now, for many paleontologists working today, one of the most important tools they can use to study fossils is CT scanning. And that is using powerful X-rays to look inside the fossil and reveal its structure. So I'd like to tell you more about that today, how CT scanning works, how we gather the data, and the way that CT scanning has transformed the kind of information we can gather about extinct animals and the processes of evolution. So I'm sure you've all heard of X-rays, and lots of you have heard of CT scanning. So the CT in CT scanning just stands for computed tomography. Most people hear of it when they or someone they know goes into hospital to get a CT scan, like this guy here. Perhaps they've broken a bone or they have some kind of other medical condition, and the CT scanner uses X-rays to see through the tissues of the body and inside. And it lets doctors see organs and bones and to help diagnose problems and illnesses so that they can improve their treatments. And when you get CT scan data done of a human, it would look kind of like this. So you can see on the left-hand side, you end up with 3 slices through your object, your human body. And they're like 3 photographs, and they're taken on the X, Y, and Z axes, here color-coded. In other words, it's like a visual slice through the body, vertically, horizontally, and depth-wise. And using this, doctors can pinpoint a location in the body and they can see what's going on. So it's a really revolutionary technology for medicine. But paleontologists can also use this technology to scan their fossils. This is the skull of an extinct marine reptile from the time of dinosaurs, and it was scanned so that it could be studied by a couple of friends of mine, Dean Lomax and Nigel Larkin. Now, this is a really good example of a fossil that was found a long time ago, so over 60 years ago, being restudied using this new technology, because CT scanning wasn't invented at the time that the fossil was first found. And that's why CT scanning is so important for modern paleontologists, because the technology didn't exist until relatively recently. And now it allows us to look inside fossils and see their internal structure. We can find out more about them than we could in the past. So before we had CT scanning, what do you think people did if they wanted to know about the internal structure of fossils? Well, the history of CT scanning starts here. This weird looking contraption is in the collections at the Oxford University Museum of Natural History where I work. It sort of looks like a cross between a blender and a microscope, but it's actually called a serial grinding machine. And now it was invented by the man you can see in the top right of your screen, William Johnson Solis. Now, Solis was a geologist and an anthropologist. He was professor of geology at the University of Oxford at the beginning of the 20th century. So he studied rocks, but he was also very interested in fossils. Now, as a geologist, Solis would have been familiar with something called thin sectioning, which we still do today to study rocks. So a thin section is when you take a rock like this and you cut a very thin slice of it, and then you grind that slice down until it's so incredibly thin that you can actually pass light through it. And then that way you can examine it under a microscope. So here's an example of a thin section. And you can see all the little grains of rock that make up this larger piece of rock. Now, thin sections are really important because they allow geologists to study the composition of rocks. So what are they made of and how did they form? And if you think back to the image you saw earlier of the CT scanning in the hospital, it's actually a very similar idea. You can see through an object, in this case a rock, in a single slice, and you can study its structure. Now, people realized a long time ago that this same process used to study rocks could be used to study fossils. And it's something that we are still doing today. So here's a recent example. This is me a couple of years ago. I found this large fossil bone on the island of Egg. You can see it in the bottom of the screen there, just underneath my knees. I found this in the Isle of Egg on the west coast of Scotland. And when we found it, our team wasn't really sure what kind of animal it belonged to. We knew it came from Jurassic rocks and that it was the right kind of shape to be a dinosaur bone, but the bone was quite badly damaged and it was very difficult to identify what animal it belonged to. So my friend Greg Funston at the University of Edinburgh, he made a thin section through this bone so that we could study the structure inside it. Now, the inside of bones is shaped differently depending on things like what kind of animal you are, how old you are, whereabouts the bone is in your body, and how fast you grow. So we were able to look at this bone and see from the structure that it had belonged to a relatively young dinosaur that was still growing. And we matched the bone structure with a group of dinosaurs that includes Stegosaurus, which you've probably heard of. It's the one with the armor plates on its back. So we use this thin section to identify the bone as belonging to a young stegosaur-like dinosaur that lived and died in Scotland 166 million years ago. So thin sections are quite a powerful way to get information from your fossils. But they only give you one slice through an object or a fossil so that you can see what's going on at that one spot. And what researchers realized is that if they could cut multiple sections through a fossil, they could use that to see the structure at multiple points. So that's what Solis's serial sectioning device, that's where this comes in. So with his colleague, Solis built the device, and it allows you to mount a fossil and to carefully grind away at it a millimeter or two at a time. Then when you do that, you draw or photograph what you see, and then you grind away another couple of millimeters and you draw what you see. And then again, and then again. And you just keep repeating this process, drawing or photographing each cross section or slice through your fossil until you reach the other end. And in that way, you can create a collection of snapshots through the fossil, and you can use these to recreate the internal structure. So it's a bit like this model here, which is used for teaching human anatomy, except that when you serially grind a fossil or object, your sections are much, much closer together. But it's the same idea. You can use this to trace the bones and other structures through the slices and study them in more detail. And Solis used his sections kind of similarly to this model to recreate wax models of the objects that he was investigating. So for example, William Solis's daughter, Igerna, she was also a scientist, and she used her father's serial grinding device to study the skull of this animal. It's a dicynodont called Dicynodon. Now, Dicynodon is one of our ancient relatives, and she got hold of its skull, and she carefully ground away at it using her father's serial grinding machine, taking images like this, which is from the paper that she then published on it in 1913, charting her discoveries. So the bones in this image are pale, creamy in color, and the rock is black. And using these images, she was then able to trace how these bones fit together inside the skull, and importantly, how the bones at the base of the skull and around the ear were arranged. Now that's important because the ear bones are particularly interesting in the evolution of mammals. Mammals have very complex, sensitive ears and hearing, and we want to understand how that evolved. So her work was very important, and it showed how useful the Solis machine was. Now, this serial grinding machine was a fantastic invention, but you might have spotted a bit of a problem with it. In order to get the information from the fossils using the grinding machine, well, you have to grind the fossil away in layers. You destroy the fossil. Now, Solis was extremely enthusiastic about using his invention as a way to get information from fossils, but as you might imagine, museum curators were a lot less enthusiastic about having all of their precious fossils ground into powder. Some fossils are just too rare or special. It would have been absolute madness to grind them, no matter how amazing the results might be for researchers, because once they're gone, well, they're lost forever. So this is not a process that you could use on every type of fossil. So the answer to this problem wouldn't arrive until after the 60s and 70s. So a South African-American physicist named Alan MacLeod Cormack and an English electrical engineer called Godfrey Newbold Hounsfield, they developed the CT scanner for use in medicine. And they actually won the Nobel Prize for it in 1979. Now, this machine works using X-rays, which are part of the natural electromagnetic spectrum. So electromagnetic rays are all around us all the time, and we use them constantly. We see invisible light, which is part of the spectrum, and other parts of the spectrum include radio waves and microwaves, which are at the low end, and then X-rays and gamma rays, which are at the high end. So scientists already knew that X-rays, which are here, they could pass through objects and could be used to look at bones under our skin. But an X-ray can only give you a two-dimensional picture through the body. In other words, you could only see one axis at a time, one slice. Now, Hounsfield, he realized that X-rays pass through different types of material differently. So the rays pass through air really easily, water less easily, and bone even less easily. Now, this property of how easily these waves pass through materials, it's called radiodensity. And the CT scanner used the radiodensity of different parts of the body to basically take pictures through the body. So you can calculate the radiodensity and create images which reveal the internal structures of the human body. This is really kind of the same idea as the serial grinding machine, but of course with one really important difference, which is that CT scanning didn't involve grinding anything to dust and destroying it. So the scans could see through materials without harming them, so it was safe to use on people. Now, it wasn't long before paleontologists realized that they could use medical CT scanning to study their fossils. And not only could they look inside those fossils, but they could discover things that no one could see before, and they could study rare specimens without destroying them. Now, the first fossils were studied like this in the 1980s using medical CT scanners, but nowadays paleontologists use what's called a MicroCT scanner, like this one, which is at the University of Bristol. So the micro in MicroCT scanning, it means that this is much more powerful than a medical CT scanner that you get in a hospital. So it can look at things that are much, much smaller and more dense. And it uses more powerful X-rays, so it's not safe to use on people. And that's why you'll see on this machine, there's all these big stickers on it warning you about radiation. Now, it looks kind of like a big microwave. There's a sliding door in the front with a window in it. And this is where the fossil goes in. The walls are really thick and they protect whoever's using it from harmful X-rays that are going to be flying around inside. So we work the MicroCT scanner using the computer, which you can see beside it here, and that's where we can also see the images of our fossils. So this is a typical scanner used to study fossils. As I say, this one's at the University of Bristol. And then here's a slightly bigger version of the same thing, and this is at the Nikon facility in Oxfordshire. Now you can walk into this one, as you can see here, but you can only go in when the X-rays are turned off because it's not safe otherwise. Believe me, the inside of one of these machines is not a place you want to be. So you might be wondering, how does it actually work? Well, you place your fossil inside on this turntable. And it is just like a microwave, really. And then once the door is safely locked shut, you can switch on the X-rays. Now these pass through the fossil and they cast a sort of shadow, a bit like you make shadows in front of a light bulb onto the wall. And the shadows are detected at the back of the MicroCT scanner by a panel. Now it's just out of view in this photo, so I've just drawn in a blue box to kind of illustrate it. So this detector panel sends all of the information back to the computer. Now, because the fossil is on a turntable, you get images as the fossil turns 360 degrees in a full circle. And it's this that lets us look not only at single slices in one direction, but in all three axes, X, Y, and Z. The computer can calculate how all of the different slices fit together, and then you can make a three-dimensional reconstruction. In other words, you can make a digital 3D model of this fossil, and you can study it. So let's take a look at how we use MicroCT scanning to study a fossil. This is a fossil jaw we found on the Isle of Skye in 2016. You can see it's quite small, only a couple of centimeters across, and it's still encased in the rock. So quite often we don't actually remove our fossils from the rock. Instead, we just chip off as much of the rock as possible from around it, and then we scan the fossil. And there's two reasons for that. One is that when you remove a fossil completely from the rock, sometimes it can be damaged, and we don't want to do that. And the reason that we remove as much rock from around it as possible is that the less rock you have, the better a scan that you can get. You can see more details because there's less rock blocking the X-rays before they hit the fossil. So we took this fossil to a MicroCT scanner and we scanned it. And what I got back from the CT scanner was this kind of data. Lots and lots of pictures through the fossil. So this MicroCT scan produced 4,000 pictures, each one a slice through that tiny little fossil. And the number of pictures that you get back varies depending on, well, lots of different things, usually the size of the object. and the different settings that you choose. All of these slices are in one direction, like slices, through a loaf of bread. So the next step is to take these 4,000 images and put them into a computer program that will let me turn them into a three-dimensional object so that I can study it. Now, there's lots of different softwares that people use to look at CT data. One of the ones I use a lot is called Mimics, and it is also used by medical scientists. So this is what it looks like when you're working in it. And you can see there's 4 windows. These show me a slice through my fossil, any slice I choose through my fossil, in the X, the Y, and the Z axes. So in that 4th window is the 3D reconstruction that I'm working on. And I use tools built into this software to find the fossil inside the rock and highlight it. And this is a process that's called digital segmentation. Now, remember I talked about radio density, so how easily X-rays pass through different types of material. Well, those values are sort of represented here in this graph, and we call them threshold values. And you can use them to make quick calculations in this software about what parts of these data are the bone and what parts are rock and other materials. So I segment the fossil in one of these windows using the threshold values that I have worked out and the built-in tools in this software. And then the software calculates the bits I've selected in all 3 axes, and it computes this information and makes the 3D object in my 4th window. Now, once I've gone through all the slices and identified which bits are which, I can then create a digital reconstruction of the whole thing. And I use that to study the fossil itself. So here is that fossil jaw that we found on the Isle of Skye. And you can see that using the MicroCT data, we are able to completely remove the rock from round about it and look at the fossil from both sides. And I can also use these data to remove the jawbone and look just at the teeth. And when I did that, I found out that there were actually more teeth that were inside the jaw and they weren't even visible on the surface. And this is the power of CT scan technology. It lets you see things you wouldn't be able to see if you were just looking at the surface. And those teeth inside the jaw were replacement teeth. So that told me that this jaw belonged to a small mammal that was still very young and was just about to lose its baby teeth and grow in its adult teeth. So it belonged to an animal that might have looked maybe a bit like this. It's called Wariolestes, and it was about the size of a big hamster. And it lived in the Jurassic alongside dinosaurs 166 million years ago. Now, the cool thing about having a digital 3D model of the fossil of Wariolestes is that it meant that I could actually print it out using a 3D printer. And I printed it out much bigger than the original fossil so that it was easier to look at the details of the teeth and the bones. And I can also, of course, take this to schools and to talks and let people handle it and see this. I'm studying a lot of different mammal fossils like this from the Isle of Skye. Here's another one. This is a fragment of jaw, and it was so small that you can see there on the left-hand side, it's almost impossible to see it in the rocks among all the limpets and the barnacles. But again, I can remove this digitally and then study it in more detail and even reconstruct it. And I used that and combined it with other specimens, other fossils of the same animal that we'd found at another time, and was able to recreate an even more complete jaw so that we could study it in more detail. And often the fossils we find are barely visible at the surface. This is another example here, but we now know that if we collect them very carefully and we MicroCT scan them, we often find that there's a lot more hidden beneath the surface. So this is the scan of that fossil that you can see, and only the tip was visible in the surface. But again, thanks to the power of X-rays, there's no need to try and remove this fossil from the rock because you can just see through the rock. And it's just as well because it could be badly damaged. In the past, very small fossils like this, it's only 1 centimeter or 2 across, they would have been removed from the rock by dissolving the whole thing in acid. And this meant that the fossil often broke into lots of small pieces and got jumbled up. And we lost a lot of information by studying fossils in that way. And it's not just jaws that we're finding on Skye. We have other fossils, including partial skeletons of animals, mammals, but also other animals like reptiles and salamanders. So this is an example of a partial skeleton belonging to a mammal called Borealastes. So this is another small mammal that lived in the undergrowth and the lagoons around Jurassic Scotland. And we didn't know much about it before now. This skeleton, which you can see scattered on the surface of the rock, well, this was found in 1972. Now, I've added the bright colours to the different bones to help you see them. But the problem is that when this fossil was found, nobody could really study it because it was so small and most of it was still stuck in the rock. But by scanning this specimen, we could look inside and through the rock and see the bones that were still encased there. You can see there's quite a few more of them under the surface. Now, one of the bones is this one, and it's called a petrosal bone. And this is found at the back of the skull. You can see here modeled on a skull where that bone would sit. Now, it's the bone that holds the inside parts of your ear, where it goes into your skull. And if you remember what I said earlier on about ears, I mentioned that the evolution of mammal ears is a really important part of understanding how the whole group, how our family group developed and evolved over millions of years, because we have this really sensitive hearing compared to other animals. So this bone is really important, and it tells us a lot about how modern mammal ears evolved and how we got our hearing. Now, when I MicroCT scanned this bone in Borealastes, I was able not only to reconstruct the bone itself, but to trace the pathways of all the nerves and the blood vessels. So in this image, you can see the front and the back of the same bone. And inside, the orange color, that's the shape of the ear organ itself, sitting inside the bone. Now, snaking all around it are those blue blood vessels, winding in and out of the bone and supplying the ear with blood. And the yellow thing, well, that's a bundle of nerves in the ear. Now, this whole bone is only a couple of millimeters across, so it's smaller than a grain of rice. But the power of X-rays makes it possible to see incredible detail and see through this fossil, all without damaging it in any way. So this sort of information would have been impossible to imagine for somebody like William Sollas when he invented his grinding machine back in the early 1900s. So you might be wondering, where do we go from here? Well, of course, this technology, it continues to advance. So I said earlier that MicroCT scanning was an even more powerful version of regular medical CT scanning. Well, there is something even more powerful again. It's called Synchrotron scanning. So here's an example of a synchrotron facility. This is the European Synchrotron Radiation Facility in Grenoble in France, where I've scanned some of my fossils. Now, synchrotron scanning also uses X-rays, but these are created using electrons, which spin around this giant white ring you see here. Now, the X-rays that the synchrotron produces are 10 trillion times brighter than the X-rays used in hospitals. Now, because they're so much more powerful, we can use them to look at much, much smaller things, including the microstructure of materials and even the motion of atoms. We can also use them to look at fossils. So synchrotron can reveal details in fossils that conventional MicroCT scanning, the stuff I showed you earlier, just can't manage. So we have a lot of fossils from Skye that have been scanned with the synchrotron now, and in the next few years, I'll be able to share with you more and more of the discoveries we're making using this powerful X-ray source. But in the meantime, here's an example of the kind of things that we can find out using synchrotron. This is an image taken at the synchrotron by my friend Ellis Newham. It looks a bit like a slice through a tree trunk, yeah? And you can see there's rings all around it. Well, it isn't a tree trunk. What you're looking at is a slice through the root of a 200 million year old mammal tooth. The tooth is only a millimeter or two in size, and it belonged to a mammal from right back at the point where mammals just started to look recognizably like mammals. They were small, they were mouse-like, and they hunted insects by night. Now, we don't know very much about their lives other than what we can see from their teeth and bones. But this image, which is taken using synchrotron X-rays, tells us something really startling and new. Ellis and his fellow scientists could count the rings in this tooth, just like the growth rings in a tree trunk. So each ring is roughly 1 year of growth. And so by counting these rings, they could actually find out how old this tiny 200 million-year-old mammal was when it died. Amazingly, they discovered that this tiny creature lived to be 14 years old. Now, I don't know if any of you have ever had a pet hamster or gerbil or mouse. Well, they only live for 2 or 3 years at most. In fact, most small mammals today don't live very long at all. And they certainly don't live for 14 years. So these ancient mammals, they must have been quite different. They must have had different metabolisms. That's like the rate that their bodies produce energy. They might not have been quite as warm-blooded as modern mammals are, and their life cycle could have been completely different as a result. So this gives you an idea of how completely discoveries like this, how completely synchrotron and X-rays can change our idea about evolution and about science. And hopefully in the next few years, we can start using this sort of technology to look at more of the fossils from Skye in greater detail and tell you more about how they lived and how long they lived for. There's a lot more to be discovered on Skye. And one thing is for certain, that using X-rays and CT scanning has changed the way that we study extinct animals of all kinds. Thanks very much for listening. I'd be delighted to take any questions.